Thank you for the invitation to present my work in this meeting today. So the idea is to give you a short overview of the work done in my team, which is focused on, as it was introduced before, the introduction of different single-cell data modalities in order to better understand the cell behavior. So first of all, I don't know why, yeah, we see a 53 in the middle. So with the introduction of single-cell sequencing, more or less 20 years ago, it started to be possible to measure different data modalities at the resolution of the single cell. So we can now extract from one single cell different types of information, going from gene expression to DNA methylation, chromatin architecture, and spatial information. I hope the animations work. And all these different data modalities are providing complementary information on the same object that is the cell. So it's like if you're looking at the same object from different viewpoints. And it is from this aspect, the complementarity in between all these different data, that comes the interest to be able to jointly integrate them in order to better understand the cell behavior. We don't see it. So the focus, sorry, is it possible to connect my laptop? Because I think that like the presentation is totally disrupted. And yeah,
I guess so.
Do you think it would be possible?
This is your laptop?
It's a bit better. So the idea is that with the question at the methodological level that we try to address in the team is to develop multimodal learning methods that are able to jointly learn from all these different data modalities in order to achieve a comprehensive view of the cell. And this requires to address different challenges. With this one, I'm moving that one. Different challenges. The first one is that we are dealing with data that are huge. And so we have a setting where we have coupled computational complexity with large size. We have tens of thousands of features and millions of cells. The second challenge is that the data are dramatically sparse. We are with single cell data that have 80–90% of zeros, and they're dramatically heterogeneous. Some data contain pinks, some data contain genes, and it's not obvious how to map them together while keeping the full richness of the information contained in the different data types. And finally, we are in an alien supervised setting. While in the context, for example, of image analysis, frequently we have some labels of annotations of the data on which we can train a model. In this setting, we don't know the cell types, the cell states that we want to identify. And so we have to discover it from the data. We don't have labels that we can use to train the models. And the question that we try to address in my team are at 3 levels. The first one is to try to resolve cellular heterogeneity. So develop methods that integrate different data to identify the cell types and cell states that are present in the data. The second question is to dissect what are the molecular mechanisms that drive this heterogeneity. So what interactions in between genes are making one group of cells different from another one. And finally, to predict cell dynamics and trajectories. So to be able to integrate these different data types in order to reconstruct the temporal evolution of our cells, how they modify themselves at the phenotypic level in presence of different stimulations. I will start from the first direction. In the first direction, we have been deeply collaborating with Gabriel Perrier from NS, which is a mathematician expert of optimal transport. And the idea here, the objective is to be able to design models that can jointly integrate in the same latent space omics data profiled from different cells that are complementary. So we want to project them into a shared latent space where we use the complementarity in between the different data types to better understand the heterogeneity of the cells. And at the same time, we want this integration to be driven by the biological signal and not by the technological differences that exist in between the different data modalities. So for this, we have been introducing the use of optimal transport for measuring similarities in between cells. That's the PhD work of Cantini in my team. And the first step is that we can see cells in our single cell data, for example, single cell RNA, as probability distributions. And we can measure their distance in order to determine which cells are similar to each other using optimal transport that will evaluate what's the minimal effort that we need in order to morph the gene expression distribution of cell A into the gene expression distribution of cell B. Why this works particularly well? Because in presence of dropouts, so this sparsity of single cell data that we have and we discussed before, especially in RNA-led data, in this kind of approach, given that the cost that is associated to genes that have a similar behavior across cells will be close to each other, we will detect a more close similarity between cell A and cell B, even if one gene that is similar to another one has a dropout into its expression profile. While if we use, for example, Euclidean distance, as is standardly done, if a gene presents a dropout in the evaluation of the distance in between the cells, this will have an high weight into the definition of the distance. So just to give you an overview of the performances, if we take the standard clustering algorithm, SERA, at the time was the most used one based on KNN, and then latent clustering of the network. These are the performances that we obtain in different data modalities using Euclidean distance, which was the standard approach using SERA. Now, if we use exactly the same algorithm, but we replace optimal transport in place of Euclidean distance, that's the improvement that we can observe. And it was particularly interesting to see that in all cases, optimal transport was able to better detect similarities in between cells. Then we try to challenge it more in the integration of different omics data. And we work first on paired multi-omics integration, so different omics data profiled on the same set of cells, as we can get with NX Multiome or CytSeq technologies. And the more challenging integration of unpaired data. So the case where you have different data modalities, for example, RNA attack proteins obtained from different cells in the same biological condition. And here you want to integrate the data while you don't want to convert the features of the different data set to the same elements, and you have different cells, so you don't have anything that is mapping them together. I will focus on this second direction, and this is the PhD work of Jules Samaran, PhD student in my team. What we did is to develop a C-confluence, which is based on regularizing inverse optimal transport. So basically, first of all, the method is trying to address what most of the integrative methods do, that is reducing the dimensionality of the two data types. In this case, for example, single-cell RNA, single-cell attack, we use autoencoders to project them into a lower dimensional latent space. This transformation alone is not sufficient because it's just reducing the dimensionality of the data, but it's not learning how the cells are related to each other. And to learn how the cells are related to each other, it's where optimal transport enter into place. So what we are doing is to use one translation of one data set in the form of the other. For example, translating a single-cell attack into gene activity, which is a noisy transformation of attack into single-cell RNA. And in this space where both data sets are composed of genes, we can compute their optimal transport distance, identify a transport plan P in between these two data sets. And then we can impose this transport plan P in the latent space, which will help us reposition the cells in the latent space accordingly to this. So this means that we use the full richness of the features present in attack data for the projection and for learning their latent representation. And we just use the translation in order to learn how to replace, how to position the cells in a way that is informed by both data modalities. We then benchmark the method, and here you have an idea on two data sets of the performances. It's Open Problem Multiome and bone marrow CytSeq. The purity will tell you to which extent we tend to put close cells that belong to the same cell type. Transfer accuracy is coming from the fact that we are working with data that are from the same cell. We are not using this information in running the method, but we can use it to evaluate the quality of the integration. And we can see that if we go to the closer cell to an RNA cell, for example, we will find rapidly the corresponding cells from the attack. And then connectivity, which will evaluate to which extent the two modalities are mixed, and FOSTTM, which is also evaluating to which extent there is a good entropy in between the two modalities, and closer it is to zero, better it is. So you can see from this evaluation that SC-confluence performs on the top with respect to the alternative methods, and it is particularly advantageous when you work with data that have few features. So if you want to integrate a data set that is huge with a data set that contains few features, as in this case with genes that are 20,000 and proteins that are few hundreds, there is a particular advantage in this type of approach that doesn't require to translate the features of one data set into the other. Just to give you an example of an application, here it's an integration of single-cell RNA and SNFish. So single-cell RNA, the cells are dissociated and then sequenced. With SNFish, instead, we don't dissociate the cells, it's imaging-based. So based on a fluorescence value, we can assess the expression of genes. We can measure way less genes, but we can measure them in space on a slice of tissue. Here you have the results of the integration of the two data types, and you can see that we can well recapitulate all the populations that are present in the mouse cortex. But then we can do more. Given that we have the spatial distribution of gene expression, we can try to start from data in the single-cell RNA and predict how they would look like in the SNFish. So what would be their expression profile in the tissue, their pattern of profile? On the top, you have an example of 3 genes that were present in our SNFish dataset and that we hide to the method. And then we test the quality of the imputation done by our method. We do this recursively, randomly selecting 30% of genes every time. And here you have an overview of the behavior. So you can see that let's say at the macroscopic level, the pattern of expression is really similar in between the imputation and the ground truth, with some differences. For example, in the second gene, here we have a downregulation in this area that is not captured by our imputation. Sorry. And then we can go further and take, given that in single-cell RNA we have 20,000 genes, we can take all genes that were not measured in the SNFish and try to predict their expression profile in space. And what we could identify are some genes that were not present in the original SNFish experiment, so they were not identified as markers and possibly relevant genes, that were having instead the same patterning of some of the marker genes that were detected in the experiment, and that could possibly be interesting for those different brain regions. Then the second direction is to dissect molecular mechanism. And for this, we have been working in different directions, integrating multi-omics data and the use of transformer models. So I'm going to focus here on the transformer model part. So we have been developing Sprint, as anticipated before. That's the PhD work of Jeremy. And Sprint is based on a transformer architecture. So it takes in input data, genes embedded according to 3 tokens. The first one is the SM2 protein embedding of the gene. This is substituted to the gene symbol in order to be able to translate easily the results of the model to other species and other settings. The gene location of the gene, we want the model to know that genes that are close to each other might influence and regulate each other, and the expression values of the genes. And then all this is given in input to the bidirectional transformer, which is trained according to 3 main tasks. So we have 3 losses according to the 3 tasks that are summed up together. We wanted to do something different than what was done by state-of-the-art foundation models for single-cell RNA that are just using masking. So they took what was available in natural language processing and applied it directly to single-cell genomics. Here we wanted to have pre-training tasks that make sense with respect to the biology of single-cell RNA and the biology of genes. So we took into account denoising. We know that single-cell data present dropouts, that different technologies brings to different let's say, batches and technological differences in between data. And so we want the model to be able to take a noise profile of gene expression and reconstruct its original values. Then bottleneck learning. We want the model to be able to start from the full expression profile in the cell and restrict it to fewer dimensions, and then reconstruct back the full expression profile of the cell, a bit in the spirit of the autoencoders-based models and all what is done in single-cell RNA in embedding gene expression data. And then classification. As we have available in our data a lot of labels for these cells, we want the model to be able to recognize whether the cell is coming from a certain tissue, whether it's from a disease or an healthy patient, which type of cell, the sex of the patient, and so on. We can do all this training because we have available cell-by-gene data, which contain profiles for 50 million cells, deeply annotated with a lot of different labels. And then what the model can do afterwards, it can make different types of predictions. We tested in the project zero-shot predictions, but in principle, it can also be fine-tuned on additional data with better performances. So it can obviously denoise the expression values of a new cell, given that it's been trained on denoising. It can produce the convolved embedding, so identify batch effects in between different data and produce an embedding of the cells that is correcting for this. It can predict labels, so say which cell type is corresponding to a different cell that is given, which disease, and so on. And it can also predict gene network, which was the real focus of our work. So the idea here is that if the model is learning all these different aspects of the cells, it should have understood the basic interactions in between the genes that determine this decision making. And so starting from the attention matrices of our model and averaging them, we can get gene-gene interactions that are responsible for all these predictions. And we use these as our gene network associated to the prediction. We use also another approach, which is based on the use of a classifier. So in case you have a few links that you know that are present in the system, then you can train a classifier on top of the attention matrices and you get better quality gene networks. But in real biological applications, you never have interactions that are known and that we are sure about. And so we wanted to have both types of approaches available. And this is an example of an output. So you can see like an embedding of the different cells present in the data, the annotation of them based on the different cell types and disease setting, tissues, and so on, and also the networks associated with the different cell types. We then benchmark the method. First of all, we focused on batch effect correction using SCIB, which is a openly available benchmark provided in open problems. On the right, you have the methods that are not using batch labels in order to realize the batch correction. And on the left, you have those that use the batch label. So you can see clearly that those that use additional information have better performances. But among those that are not using it, Sprint, which is our method, is among the best performant one. SCGPT, for example, that is one other state-of-the-art foundation model, is having lower performances, and also Geneformer, and the SC Foundation is comparable to us. Then the other problem is classification of cells. So here you have performances according to cell classification. Again, on the right, you have the methods that are not using the cell labels for training. And on the left, you have methods that are not using the cell label for training. And on the right, those that are using it. So obviously, those on the right have better performances. But among those that are not using it and that can perform it, Sprint is the one having better performance. And then finally, on gene networks. So we have here different methods for gene network inference. Among them, Gene3, for example, which is the state-of-the-art methods for gene network inference, and different foundation models, Geneformer and SCGPT. What we see here, it's a perturbation data set. So we have a ground truth of interactions. And we can see that Sprint is performing better than SCGPT and Geneformer already in zero-shot prediction. Gene3 is, however, still better, which is the state-of-the-art if you want to infer a network on a data set. It was interesting to see that what we did afterwards was to try to use the classifier, as I was saying before, and use some few interactions that are known in order to predict this network. And it was interesting to see that if we use OmniPath, and so known literature-based interactions in between transcription factor and genes, to train a classifier, we were actually getting worse performances with respect to the zero-shot prediction. If instead we use few perturbation data to do the training, then we achieve performances that are comparable to Gene3. And this was interesting to notice because it means that different ground truths that are available for gene networks have little overlap. We know this, and that not always using known ground truth can help to achieve better performances in this field. Then the last point is predicting cell dynamics and trajectories. And for this, we wanted to take advantage of spatial transcriptomics data that start to be available at single cell resolution and on which atlases are available profiling the same system at different phases of the same biological process. For example, here development and the zebrafish during development. So in this context, we have been developing stories for spatiotemporal omics energies. That's a joint work of Jules and Gertian, still PhD student in my team. What he's doing is to start from this spatial transcriptomics data, measure the different viewpoints, and is learning a differentiation potential. So the objective is to learn a sort of Weddington landscape that can explain the temporal evolution of the cells, placing cells that have higher potential to become different types of differentiated cells at higher value of this potential J, and cells that are more differentiated and specialized at lower level of this potential J. Is doing this using a neural network and an MLP in order to learn the potential J, And what the MLP is addressing is minimizing the loss, which is computing the difference in between the prediction that we can make of what the cells from time t will become at time t plus tau, and the actual measurement that we have available at time t plus tau. So all this is based on the fact that we need to have different temporal measurements of our cells. If you have only 2 time points, it's not possible to achieve a good inference of the potential. And how is minimizing this loss, how is computing this different? Is doing this using a fused Gromov-Wasserstein loss. Basically, what we are doing is to have 2 main components in our loss. One first component, which is linear, and which will evaluate to which extent the cells are similar at expression level. So to which extent the cloud of cells that we have measured at time t plus tau and our prediction of how the gene expression will be at time t plus tau are similar to each other. And then we will have a second term, which is quadratic, which is instead using the spatial positioning of the cells. So we want that cells that are close in space in the time t plus tau are also close in space in our prediction of the time t plus tau. So we ask for, let's say, the model to have also a spatial consistency and not only based on the expression evolution. And then the output that you can get are trajectories that you can plot as velocity arrows on top of a U-map, or you can visualize through the differentiation potential. And given that explainability here is fundamental, because we don't know whether the prediction is based on some artifacts, some technological differences, and so on, we also give gene trends that are associated to the gradient of this potential and the velocity in order to be able to explain biologically the different directions and whether they make sense with respect to what is known in biology. We have been benchmarking the method in 3 atlases: axolotl brain regeneration, zebrafish development, and mouse development. And we have 2 test sets: an early test set where we are taking another batch, but from a time point that has been already seen by the method, and then a late test set where we are taking another batch and from a time point that was old out during the training of the method. So here it's making a prediction on something that has never seen at all. We are evaluating the performances based on the Wasserstein distance. Here you have stories, and the other method is prescient, which is a method based on a similar, let's say, objective of inferring a differentiation potential, but that is not using the spatial positioning. So the question here was, is the fact of using spatial positioning in the way we do with stories more informative, not than doing it? And what we could see is that our prediction were always more close to the last time point that we had to predict in all the different test cases, both early and late, and in all 3 different atlases. So this convinced us of the quality of the spatial information for inferring cellular trajectories. And then for the mouse dataset, we had also some annotations done by biologists on what are the true transitions from one cell type to the other that should happen at a different time point. And so we could compute the transition accuracy that was telling us that also in this case, the accuracy is higher in the case of stories that is using the space rather than in the case of prescient. So this is not only just a difference in terms of distance, but there is really a biological consistency of the transitions that stories is predicting. And just to give you a concrete example, here you have adaxial cells, and you see the mapping of stories from one time point to the other one, which is localized correctly where adaxial cells are localized. While in the case of prescient that is not using spatial information, the prediction is more widely spread across all the embryo. And finally, just to see some examples of biological application where the space could have a crucial role, we focused on the mouse brain and the transition from radial glia cells to neuroblast and glioblast. Basically, there is a splitting of the cells that move either to one state, either to the other state. And we were wondering whether spatial information could have a key role in this transition. And what we could see here, you have one of the slices, E16.5. What we are coloring are radial glia cells, glioblast, and neuroblast in this slice. Radial glia cells are a bit localized whole here, while the final states, neuroblast and glioblast, have specific spatial location. Neuroblast area on the top, glioblast area in the middle, then we have another bit of neuroblast here, and finally glioblast here. And here, what we are plotting is for the only radial glia cells, the fate probability inferred by stories. And what we can see is that radial glia cells that are all localized here don't randomly commit to one of the 2 states, but they do it depending on their spatial positioning. So the radial glia cells that are here on the top will tend to commit to neuroblasts that are really positioned here. Then here, radial glia cells will go to red, which means committing to glioblast, which is the state that is here. Here again, we have neuroblast, and finally here we have again glioblast, indicating that the space is playing a driving role into the commitment of radial glia cells. And without taking into account, we couldn't make good predictions of their commitment. So with this, I want to conclude. I want to thank the organizers for inviting me to present today, all my team, and in particular, Jules, Gert, and Jeremy that did all the work that I presented today, all collaborators and funding, and I'm happy to take questions.
Thank you very much, Laura. Do we have any questions? Yes.
Thank you very much. Very nice talk. I want to comment or emphasize or something on this mention that you when you included some known predictions from some data sets, from some known, I don't know what is the ground truth there, because I'm always looking for a ground truth in gene regulatory networks, that the accuracy drops. And to comment a bit, because I understand that when we include something that is not right, it drops. But what have you done after in order to improve it? So the slide that you have it?
Yeah, so you mean what was the other element that was close by, right? The other performance.
That one, exactly.
Yeah, so this is basically, it's really simple. It's just using few links that were in the ground truth to train the model and then testing it on those that were not in the ground. So then we see there that we have an improvement of performances.
So how do you just extracted randomly some?
Exactly, that we use for the classifier.
So this means that the ground truth that we have is not a real ground truth.
Yes.
You can say that.
Yes, I mean, also the ground truth that we're using for evaluation is not a real ground truth because it's perturbation and it presents a lot of indirect effects. So we have a real problem that is the need for good ground truths to evaluate gene regulatory networks, maybe based on ChIP-seq and binding of transcription factor together with perturbations, but this doesn't exist in large scale. Or maybe the networks can be evaluated in different ways. For example, based on how well they can predict transformations of gene expression profile. There is some work done by the lab of Martin Luken, where they are developing a benchmarking of gene regulatory networks based more on the effect of the cells rather than on the quality of the network itself.
Two very quick technical ones related to that. One, does your model predict directionality? And second, if you have tried to do any, apart from the comparison of accuracy or performance or whatever you measure here, I don't know, sorry, with Gini3, which is a good hierarchical method, etc. If you have tried to see the overlap, not just the overlap of the overall prediction. That's it. Yeah. Thank you.
So what was the first question? I forgot it. Yeah, so it can predict directionality. That is true. Regarding the overlap with Gini3, we try to compute the overlap. It was better than the overlap that you have in between different ground truths. But then it was still not super high, like it was among like in between 50 and 60%, which is still quite reasonable. Yeah. We have also to say that Gini3 is applied exactly to the data where we are inferring the network while we are doing zero-shot prediction. So the method has never seen those data and is making a prediction without having any information on the cells.
Yes, there is another question.
So maybe that's something I didn't understand. It's about spatial transcriptomic through time. I thought that like spatial transcriptomic was destroying the tissue. So how do you get data through time?
Yeah, that's a good point. So maybe I didn't stress it enough well in my presentation. Basically, that's exactly the problem that we have methodologically, that every time you are destroying the tissue, and so you need to have some way in order to learn what is the mapping in between the cells across time points. And that's what the method is trying to do. So it's trying to learn how the point clouds of different time points are related to each other and building this potential J, learning this potential J that is able to map cells across the different time points and also predict how they will look like in a future time point that is not been measured. Okay.
Time for a last short question.
Thank you for a exciting talk. I want to focus on the model that you try to use a MLP to model the Waddington's landscape, and then you use the gradient to try to explain the genes regulator, the role of find out the genes regulator, right? However, I want to ask the question that because of the train rule, if you have one genes A regulating genes B and then interacting with gene C, then you will have an interaction from genes A to gene C. So it is very hard to distinguish in just using the gradient-based method. Do you have any plan to resolve this aspect?
Okay.
Thank you.
So that's a good question. So at the moment, what we want to do is just have sort of marker genes that are associated to the evolution of the cells. So if there is a direct or an indirect interaction, if a gene is coming downstream of other, we don't care for the moment. We just want to say, okay, do we get markers of stem cells in the beginning? Then this seems to make sense. Do we get markers of an intermediate cell state afterwards? Then this seems to make sense. That's the level that we have at the moment. However, we are trying right now, without space, to also infer mapping in between cells together with the network of gene interactions that can explain these transitions. We are doing this alternating an optimal transport problem optimization and an inverse optimal transport problem that should help us learning the A matrix of interactions in between the genes. And so the next plan is aligned with what you are saying. There is here, however, no MLP, there is no potential, and there is just the idea of learning mapping in between cells and gene interactions responsible for that mapping.